it's really, you know, we see a snapshot of the seafloor. We don't get a chance to sit there and watch, you know, who visits corals or who visits sponges and eats them. And anytime we make these observations, it's like we expand our knowledge of food webs in the deep ocean, which are very poorly understood. So any kinds of events like that, speaking broadly, are my favorite.
So shoreside, we are past this feature that was not a feature. Still slightly headed upslope.
It is a little bit steeper, though. I've had to step up some.
Okay, great.
Not
This actually looks
significantly.
like a good spot for a scoop for Daniel. Could we pause here and do a little scoopy scoop?
Sure thing. The ship just stopped, so wherever looks good.
Yeah, we're seeing some nice shells and some smaller shell hash, so I think this might be a good opportunity.
Can you zoom in now?
It's like clamshells.
Yeah, clamshells and maybe some worm tubes.
Is that of interest?
Smaller. Yeah, let's
Maybe scaphopod shells?
I'm asking Daniel in the chat.
Or small worm tubes.
He's saying this is good biogenic material, so yes, this would make a good scoop.
Okay. Can you zoom out now? Is there any better areas here, or just in general?
Just anywhere where the density of those tubes and shells are hash or high, and it looks like right in front of us.
And this is a scoop?
Yes, please.
Okay.
Yeah.
Fill it up.
Fill it up high.
Yep. Daniel's saying full scoop, dig down and fill it up. Pretty please.
Full scoop ahead. Steve, what sample number is this?
We're on 149.
Wow. We've been out here for a while.
Still haven't broken my all-time record yet, though.
What's that record?
What's that?
It was in the 250s.
Whoa. In what region?
For how many days?
Gulf of Mexico. It was 2 weeks.
Wow.
Geyser expedition.
Wow.
Looking at seep stuff. We were doing a lot of super sampling. And the super sampler has many different...
We might need to turn off that thruster. Yeah. Yep.
What are you going to do with this sample once it's collected?
Yep, draw out.
We are going to ship that right to Daniel. Actually, he's coming to pick it up. So Daniel is with the Santa Barbara Natural History Museum, and he's looking for these really cool biogenic habitats that have pretty much what that means is there's lots of living organisms in this habitat, and you can kind of see there's lots of shells, broken shell pieces, and worm tubes. And so he wants to look at some of the really small organisms that live in between these pieces and sediment grains. He's specifically looking for micromollusks on the order of 0.5 to a couple of millimeters big. And he's describing new species and cataloging and vouchering older or already known species that live within these habitats. And we've been taking scoops for him at different depths at different portions within the Santa Lucia Bank and Channel Islands. So you never know what you're going to get. And I would say this is probably not a super well studied habitat within the deep sea. So it's likely going to produce newly described species. And Daniel's going to do this by looking at the scanning electron microscope. So he's going to look at these samples and look for these micromollusks.
Scoop likes
after
to land in
the
the worst
samples are
positions,
processed.
doesn't it? It's very uncooperative. Yeah. Yeah, I think it is.
It's some counterbalance there, counterweight.
Yeah. It's definitely in need of some engineering.
Well, the scoop technology has increased so much since I've been out here. It used to be like a cut open milk jug. Yeah, much better.
It's still not great. Like, oh, we got the drifty arm thing going on. Hang on. Exit.
I don't think I've ever seen you do a scoop. This is pretty fascinating to watch.
It is. It's cool.
Oh man, that is drifty.
We can scoop up that flatfish.
And just a top layer.
Yeah, last time he was happy if you went down a few centimeters, so maybe down to 3 centimeters or so.
All right, I gotta get a better grip. This is no good.
Looks like that fish is trying to pretend it's not there, sinking down.
Oh, come on. All right, what's the grip force at? Boy, that's not good.
I think you're going to get the fish.
Did you see that note from Daniel that he took 13,000 scanning electron micrograph images for his last big project? So lots of data will result from this scoop.
We look forward to seeing them from our own neck of the woods. We're really excited about this expanding our partnership with the Natural History Museum.
I have a couple more
Can you
questions here. Let me know when is a
give
good
me
time to ask them.
the thruster back? I need more downforce. All right.
I think you can ask another question, Laura, while they're given their scoops.
Okay, a good one just came in. Is there a website or someplace where people can see the list of all marine invertebrates that have been observed around the Channel Islands on this expedition? Listed as by genus species names, preferably.
That's really interesting. So there's definitely not a comprehensive list available as of right now in terms of what we've observed, because we were collecting transects, which will be analyzed by video analysts at a later time and annotated. But we do have our collections list, so we have maybe not everything we've observed, but things that we've collected, which are mostly sponges and corals. I'm not sure if such a list exists like that usually from these kinds of missions. So unfortunately, I don't think we have that resource for other scientists.
Well, one of our tasks is working with the Marine Biodiversity Observation Network folks. There's three, I think, they call it MBON groups around the country. We're working with them to characterize the biodiversity in and around the Santa Barbara Channel and figure out how best to do that. We're just coming up right now with our very first kind of more comprehensive species list across all taxa, and that's a project that'll take years. But yeah, we don't even know the extent of all the different organisms within our own waters. There's just so many and so much area that's still unexplored. So part of that project is to figure out what's been kept in old museum specimens, what's been documented in books that has a range that covers the sanctuary, everything from algae to whales. So we're just trying to get a handle on all that right now, and it'll be quite an undertaking.
Well, and I'm wondering if this is potentially another scientist asking who might want to review some of these videos for maybe their specific project or species of interest. So I'm actually wondering if they're asking from these dives that we've been doing, is there any list that we'll make available where, like, for example, oh, we saw fragile urchins on this dive, this dive, and this dive, you know, so they would know which videos to request potentially. But I don't think we have that information available as a resource.
There'll certainly be a cruise report that'll come out at a minimum. So we'll have the number of dives and the different samples that we collected, and that can be referred to.
Yeah,
There's
but I
a
think if
note
they're
from
looking
Daniel.
for something specific, we won't have that.
I think it's a
Looks
pretty
like
full
Daniel
bag.
says there's.
That looks great. Thank you.
You're the first person to take a full bag literally, I think.
That is great.
Just saw the note from Daniel that there is a taxonomic atlas series from the Museum of Natural History on Santa Barbara Channel organisms.
Let me see here. Okay. Pretty messy. Just a little bit. I think it's okay. Go for it. Is it closing? It didn't close all the way. Is it hung up? Might be hung up.
I know we're at time to wrap up questions here. Let me know if you're open to one or two more, or if you want to call it a day, and I'll sign out.
Yeah, we can finish with a few more questions. I'd be happy to.
Down underneath the thing yet? How about that? Try that. I think it's in there. The bag is definitely full. What's happening with the floaty ball though? Well, it might keep the lid from closing though.
A question
Okay,
from Florida
roll in the
on
box. Okay.
a question from Florida on what types of organisms samples are particularly susceptible to adverse pressure changes when returning to the surface for studying.
And before we launch into that question, Shoreside, is there any other sample that we want to collect here before continuing on?
No, I think we're good, especially if we want to start a transect.
Roger, we'll do a 200-meter transect with lasers on towards AY.
Perfect.
Great, thank you. And to answer your question from Florida, yeah, there are several species that may not make it to the surface alive with the pressure changes. But I think it just depends on what you're targeting to collect. So sponges and corals seem to do pretty okay bringing them to the surface, and we've been working with scientists that are keeping them alive in aquariums or aquaria to do growth studies, feeding studies, and it sounds like they don't need the pressure to survive. They need more specific close controls on the temperature and the oxygen and the pH of the water more than anything else. But there are definitely organisms that are affected by the pressure changes, and that if you want to keep them alive for aquaria purposes, they need to be brought up in a chamber that kind of keeps that ambient pressure on them. And I think ctenophores might be a good example, although they're kind of gelatinous, so maybe that's not a good example. We had been talking a little bit with the Monterey Bay Aquarium during the West Coast Deep Sea Coral Initiative, and they shared some of their insights as to trying to keep some of those deep sea species alive, to kind of test out what's possible to keep in an aquarium for an exhibit and what's not, but not necessarily for all of the organisms is that high pressure necessary for them to live and survive, but it is for some.
In the lab that I belong to, we've had really good success at keeping Lophelia alive for several months. Actually, I think we still have Lophelia from over a year ago in the lab living right now. Most of the time, the Lophelia we collect is shallower than 500 meters, 700 meters tops. But we've noticed that Lophelia collected much deeper than that, or even animals generally collected much deeper than that, like sea fans around 1,000 meters and deeper don't last as long, maybe a few weeks to a couple months tops. It's not clear why that would be the case.
Right.
But there's also some effective pressure on protein folding that could be why the organisms can't persist for as long. Temperature and pressure on molecular structures in the cells of the organism. But we're talking much deeper, several thousand meters.
Yes, yes, and soft-bodied organisms too, like some of the fish, or once you bring those up out of pressure, they almost rely on that pressure to keep them solid at those really deep depths. So when you bring them up to the surface, where the pressure is much less, it's just they get kind of flabby almost, like they just can't keep their shape anymore.
I would imagine it's harder with animals with air spaces like swim bladders and things like that to bring them up.
Yes, that too. So rockfish too that are caught at generally much shallower depths than some of the places we're exploring, but when you bring them up from several hundred meters, their eyes and their swim bladders will expand and bulge out, known as like a decompression issue. So if some of them make it, in order for them to survive, they have to be taken down at that pressure because now they're too floaty essentially to go back down on their own. And there are devices that are triggered by pressure. I think this is another box grab here on the left. So you kind of attach it to the fish, and then it's attached to a weight, and you send it over the side, and it sinks. And then once a certain amount of pressure triggers the latch to open, the rockfish is free to swim off, and the pressure has their swim bladder and their eyes kind of go back to normal. So there actually is a pretty good survival rate of that, rather than just leaving them at their surface where they're pretty much guaranteed to perish if they're tossed over the side again.
Well, thank you all so much for answering all these questions today. Really appreciate it. A lot of grateful people out there who love this interaction. And so, yeah, thanks for taking the time to do it, and good luck on the rest of the expedition.
Thanks so much, Laura. That was great.
Thanks for moderating. Yeah, we enjoy doing this. And we are on transect now, too.
We are indeed. 30 meters in.
So if you're watching at home, when we say we're on transect, we're covering a distance of about 200 meters that we'll survey. We won't be stopping to spend time checking anything out over the course of that 200 meters or do any collections. We are just trying to get to be able to quantify, to be able to count all the different organisms, animals, sponges, fishes, and corals that we see over that distance. So we have a point for comparison with other areas that we're surveying in the sanctuary and also other areas we're surveying on similar missions up and down the whole West Coast. This work, it was mentioned earlier, is part of the West Coast Deep Sea Coral Initiative. It's funded largely by NOAA's Deep Sea Coral Research and Technology Program, but also this cruise was funded in part by Office of Marine Sanctuaries. And we're partnering with our colleagues up and down the coast to do similar surveys of ROVs, like you're seeing now, the video you're seeing now, to do mapping, sonar, collect sonar data on the seafloor to look at, identify where certain features of interest might be that congregate or aggregate these different species we're interested in, as well as autonomous underwater vehicles, or AUVs, which are not tethered like these are to the ship. That work's done. Those surveys over the last few years have been done as part of a really broad coalition of groups that have been brought together under something known as the Express campaign, and it includes key partners with the U.S. Geological Survey, the Bureau, who are interested in looking at submarine hazards, the Bureau of Ocean Energy Management, looking at areas for renewable energy up and down the coast, the Marine Fisheries Service, interested in better understanding fish habitat associations and stock abundances of different commercially harvested species. Ocean exploration research, another part of NOAA that's interested in exploring places that haven't been seen or discovered before. And the Marine Sanctuary Program, that's us, interested in better understanding, characterizing, and managing our special places. So up and down the West Coast, there's 5 marine sanctuaries ranging from Olympic Coast National Marine Sanctuary off of Washington through to Cordell Bank, Greater Farallones, Monterey Bay, and Channel Islands National Marine Sanctuary, all of which are located off the coast of California. These areas are protected by the federal government. Our specific sanctuary that Lizzie and I work for, the Channel Islands Sanctuary, manages the waters 0 to 6 nautical miles off of the 5 northernmost Channel Islands in Southern California. And largely one of the main prohibitions there is really just oil and gas exploration. So protecting this area from activities associated with oil and gas exploration. But there are a number of other rules and regulations. We have a network within the sanctuary of no-take reserves, where approximately 20% of the sanctuary is protected from any kind of take activity. But otherwise, we're largely open and accessible to all manner of users, from whale watchers to recreational and commercial fishing to scuba divers, kayakers, and so on. Collecting the kind of data that we're collecting here is incredibly useful and valuable for a number of reasons, resource protection management reasons, knowing where key or important habitats were, should there be some kind of impact, a ship grounding, a chemical spill, an oil spill, anything of that nature allows us to know where are the habitats that we most want to protect, if that's an option, or to better understand what's lost in the case of an impact assessment. We'll also get requests for permits for entities that want to do an activity that might disturb the seafloor, and so understanding where regions where it might be okay to disturb the seafloor based on the habitats present versus areas that we want to protect because of some of the species that we see there. Right now, a large portion of this dive is over soft bottom sediment. We see fewer organisms and species in this region, but we see unique and different organisms in this region than we see on the hard bottom areas. So we can't spend all of our time on just hard bottom reefs because we'll miss key habitat for things like some of the flatfish that you've seen here, some of the sea pens and other species. There's a basket star that was in the middle of the screen there, and some pink.
Yeah, we've seen a few of those.
Sea urchins. I wonder all of a sudden, Lizzie, do you think we've just come up shallow enough to see them, or
I don't know. I thought they were deeper, honestly.
Yeah, actually, they are. They are.
Yeah.
Right. Pretty deep spot. Yeah, I don't know what's changed.
Are they lost? Such bad visibility, they don't know where they are. But yeah, I always thought of basket stars as being deeper rather than shallow, so I'm a little surprised to see them at these depths. But I'm also no expert, so.
So this will be the final dive of this cruise before the vessel heads back to port. We'll be pulling here at roughly just after 3 p.m. Pacific time, pulling the ROV out of the water, get it back up on the boat by 4 so the vessel can head into San Pedro down there, LA Long Beach area. This has been a multi-day week cruise that started up in, well, combined with the cruise before it, started up off of Northern California in Greater Farallones and Monterey Bay National Marine Sanctuary, off of Davidson Seamount and Santa Lucia Bank area off of Morro Bay, part of a nominated National Marine Sanctuary known as the nomination package named the Chumash Heritage National Marine Sanctuary, encompassing the areas off of Morro Bay and around Point Conception, where the California coast turns east-west before you get to Santa Barbara in Southern California. And lastly, it abuts the Channel Islands. And now, at this point, we're in the Channel Islands National Marine Sanctuary. Lots of urchins
different kinds of
scattered.
sea
You see these urchins scattered sparsely throughout the seafloor. But what happens when some kelp or algae from the shallow waters drift down to these deeper water environments, they all can sense it and stumble across it. And you'll see just huge masses. The other day we saw a giant pile of 30, 40, 50 of the urchins all in one spot. When we zoomed in, they were all devouring a kelp holdfast that had drifted down from shallow waters into the deeper communities. So they depend on these kind of spurious, sporadic events where the food drifts down from above to feed. really getting a sense of the current with how all those how they're leaning over so much.
Yeah, and you can follow particles as they flow through the water column in that same direction that the sea pens are leaning. It looks like those basket stars are just they're hanging out on top of some hard bottom habitat. They're on those rocks. Like wherever you find a rock, sometimes you'll find these basket stars. It's interesting.
I wonder if they recruited to those exact rocks when they were really tiny.
Nowhere else to go.
So we've got about 50 meters left of this transect.
Excellent. Thank you for the update. We're seeing a couple of different species of sea pens. We're seeing these long, white, thin sea pens more frequently now than we were before, and some of them almost have like a curl to them, like a spiral. And we're also seeing the thicker Pentatula phosphorea species, but I'm not sure what species these smaller white sea pens are. I think we would need a zoom. And even if we did zoom, I'm not very well versed on my sea pen identifications.
So shoreside, at the end of this transect, will we want to take any samples or continue on our way to AY?
No one has spoken up yet in the science chat. Oh yeah, actually, yeah, that's true. I would like a Niskin here since we encountered a lot of sea pens. I think Meredith might be interested in the eDNA part here. Otherwise, we can keep going.
Any reason for a sediment core, Lizzie, or no?
I think if we collect sea pens or urchins for Taylor Ann,
then
we should take some sediment cores where we do that, but otherwise,
okay.
I've not seen much drift kelp yet either. But I do believe Taylor Ann is on the next watch shift, and so I'm sure we'll still be, I mean, famous last words, so far I've known. But then again, if we just found a bunch of hard habitat, that'd also be exciting, but she is on the next shift too, so she'll have an opportunity to collect what she sees fit. Otherwise, not quite yet, but I'm keeping my eye out when might be a good time to sample for her as well.
Sounds like a plan. We got 30 meters left.
Thank you. bottom. Just as the backscatter predicted.
Ground truth successful.
Yeah. It was not wrong here.
Nope. I did see in the dive. Yeah,
Mhm.
ground truth. I did see in the dive plan that cup corals are still on the list for UW, but I'm guessing we'll not be seeing many if we're not finding any rocky substrate.
True. Yeah. So there was like a spot or two that had some slightly brighter returns on the backscatter that come later in the dive if we were to make it that far. That I think since it's that shallow, we could potentially find some cup corals. That's why I left them on the list. Is there is a small possibility, but depending on how far we make it and if that's actually true that there is some rocky outcrops that's more than just low-lying relief, that yeah, we could potentially do some collections, but otherwise my hopes are not high for those.
Got it. I will temper my excitement for another cup coral search.
Another cup coral hunt.
Yeah. Cup coral quest.
Yeah. Oh, I like that a lot. Cup coral quest 2020.
To find a new exciting animal for the day.
We've seen a few octopus actually, some little guys. That was exciting. I wonder if they burrow in soft sediment. Because you said you saw one go in a hole, but I don't know if it was just because it was afraid of the ROV or if that's a thing they actually do.
Yeah, I'm not sure on the species that's common here. I mean, both behaviors are common, but just depends on the type of octopus.
Yeah. I would imagine just because there's really not any hard habitat for quite a while. So if you were to need some kind of shelter, this would make sense. And they're all fairly small.
We done yet?
Ship moves complete in 2 meters. And the transect is complete now.
Excellent.
838. Sure.
And is this where we wanted a Niskin, or do we want to keep trucking along?
We haven't taken any yet, so we can take a Niskin here.
Okay. I don't need to set down to use the arm, right? Can we, are we all the way out, Al?
Yes, we are.
Oh,
I'll double check.
yeah, it was just the, oh. Keep the trigger closed like when I first hit that. Got it. Yeah, I didn't know that that was possible to avoid.
Steve 150 for the sample.
Yep. Number 1.
Oh, that's Mongo.
Yep. That's it.
All right, good on the Niskins.
Thank you.
Anything else here that we want to look at, or we're good to go and continue once the arm gets stowed?
Good to go. Rock and roll.
Great.
Good on the arm. Am I a little bit still? Yeah. Got it. Good. All right, I think we're
Ready to go?
ready anytime.
Alrighty, we're gonna do a slight change to 176. So we land on that target. Bridge nav. 50 meters, 176, please.
All right, we can get the little junk zoom there, Al.
Ship move underway. We have 570 meters to waypoint AY.
And sure, just give a shout if there's anything we want to take a closer look on.
we want to zoom in on any of these sea pens whenever we see one, now that we're off transect? Yep.
Oh, just give me a shout. Anyone you want to zoom in on?
They're all over the place. There's one.
You're talking about the white sea pens, the thin?
Yeah.
Okay.
Yeah, they're thin, wispy coils. Any of them, really, as long as it doesn't. I don't want to make you stop too fast and say any one particular. So.
Okay, let me jut out ahead a little bit so that way we can get a quick zoom without stopping the ship.
Yeah. Visibility is bad enough. I don't want to muck it up with more dust from the bottom.
Like one of these?
Sure.
All right, Al, can we get a zoom? Oh, there's a few of them.
That's a sea panel, right?
Are we on full zoom?
I see it in my lens.
Okay.
All right, if you come wide,
That's
I can
good.
get closer if we need to. Or I do want to actually keep trucking along again.
Is there potential interest in sampling one of these, or just zoom?
I was just interested in zooming. We were talking about it on the transect.
Got it. We're about to finish a move, so we could just stop here if we want to take a closer look. Get a full zoom.
I'll leave that up to the watch leads.
Is that of interest, shoreside?
Zooms are great. So if we could get maybe one, whenever you're caught up, again, it does look like they're everywhere. So
how long left
couple
in
good
the
stills.
ship move?
This one's great. We're just one meter off.
Perfect. All right, Al, can we zoom in? Steve, let me know if there's a part of it that you want to zoom in on so I can
Just
try to.
I would say try and do a quick scan close in and then maybe one wide shot with lasers and then that's it. Some damage to the end of that colony.
Oh.
You really got to stop playing with that. You're going to go blind.
It's quite shallow, so it's really jumpy. Oh man.
Oh, maybe just a wide shot. But
Yeah.
that's perfect. All right, good captures.
All right. Oh.
Continue on. Bridge nav.
Still squared up.
100 meters, bearing 176.
Yeah, at this depth, if you just touch the pan and tilt at all, if you breathe on it, it jumps halfway across.
Yeah,
So between
176
here
is our bearing.
Yeah.
Between here and AY, there's nothing necessarily that we're specifically interested in. So however fast you're comfortable proceeding to the next waypoint, that's great with us.
Roger. We'll do a zip instead of zooms.
I'm glad that the visibility is improved though, since we're more upslope, but shallower.
Us too.
Mutual.
Do we want it?
I think we were waiting for the next watch to do drift kelp.
Got it.
We've got about an hour and 15 minutes before our next watch change.
If we do see urchins and kelp though, we can stop and grab it all since it's all near one another.
Sounds good.
But mysteriously the urchins have disappeared though.
Kelp, no urchins.
They're missing out. Where are they?